Welcome on board this vessel, the Top Notch TV. Your favorite living organisms are, as usual. Top Notch recognizes that in the modern world, there has been an increase in the number of patients suffering from or going for dialysis now and again. Today, we want to demystify the process of urine formation by first looking at the structure and the function alongside adaptations of the various structures or parts of our kidney nephron. As an organ, just like the lungs, in the lungs for gaseous exchange, we have the specific structures for gaseous exchange, that is the alveoli. And also in our kidney, we have the specific structures that will be involved in excretion, that is urine formation, which are the kidney nephrons. But first things first, we have a pair of kidneys as mammals, a pair of kidneys, and these kidneys are supplied with the blood by one of the large arteries, that is the renal artery. That is the renal artery. The blood coming into the kidney through the renal artery will be rich in oxygen, nutrients, and metabolic wastes. The main metabolic waste being urea. Then we have two other vessels that are coming from the kidney. We have the renal vein, which is removing blood from the kidney. So the renal vein will have a low concentration of urea. We have another tube that gets out of the kidney to the urinary bladder, and that is the ureter. And the function of the ureter will be draining the urine that has been formed in the kidney to the urinary bladder. But the basic functional unit of our kidney is the kidney nephron. What makes up the kidney nephron? We can talk of two structures in this, our model of kidney nephron. We have the structures that are the blood vessels, mechanism by which blood flows into the kidney nephron and out of the kidney nephron. As a branch of the renal artery, this is the renal artery, we have the afferent arteriole. The afferent arteriole is a branch of the renal artery that takes blood to each kidney nephron, which means this blood will be very rich in urea. We have the renal vein represented by blue to indicate it's a vein, and its role is to take blood from the kidney, and this blood will be having a very low concentration, if any, of urea. So in the nephron, the structures that will be involved in the elimination of urea from our blood includes, we have the cup-shaped structure, that is the Bowman's capsule. The Bowman's capsule is surrounding several branches of blood vessel, that is capillaries, And those capillaries form what we refer as the glomerulus. They are branching from the afferent arteriole. From the Bowman's capsule, we have the coiled tubes, which form the proximal convoluted tubule. From the proximal convoluted tubule, we'll have the U-shaped loop of Henle, then the coiled portions, the distal convoluted tubule, the collecting tubules. Each nephron has a collecting tubule which drains into the collecting duct. Collecting duct. During urine formation, the first process that is necessary or occurs during the process of urine formation is ultrafiltration. The processes responsible for urine formation are two. One is ultrafiltration, two, reabsorption. Ultrafiltration takes place at the glomerulus, that is where ultrafiltration takes place, whereas reabsorption occurs along the kidney nephron tubules. How does this ultrafiltration take place? This is the Bowman's capsule, and the Bowman's capsule is cup-shaped. It is cup-shaped and surrounds, or it is cup-shaped to cover very well the glomerulus for efficient collection of whatever that is filtering from the glomerulus. And during ultrafiltration, the fluid part of blood will filter out of the glomerulus into the capsular space of the Bowman's capsule. Besides the fluid part of blood, the other substances that can filter out of the glomerulus into the Bowman's capsule includes the small-sized molecules like glucose, amino acids, salts, and urea. The significance and the importance of ultrafiltration, therefore, is separation of urea from the blood. Separation of urea from the blood. What makes ultrafiltration possible in the glomerulus? One, this glomerulus comprises of several capillaries, numerous capillaries. The branching of the afferent arteriole, into several capillaries that form the glomerulus increases the surface area for ultrafiltration. We also take note that this afferent arteriole is wider than the vessel or the tube, which is the efferent arteriole that drains or removes blood from the glomerulus. This efferent arteriole, being narrow or narrower, provides resistance to flow of blood, and therefore there is kind of buildup of pressure within the glomerulus, which increases the pressure in the glomerulus, increasing ultrafiltration. But also, remember, this afferent arteriole is a branch of the renal artery. Therefore, the blood that is coming is at a higher pressure because it's coming from the heart, where pumping mechanism is taking place. So the pumping mechanism of the heart, the branching of the afferent arteriole into several capillaries forming the glomeruli, the narrowness of the efferent arteriole compared to the afferent arteriole, those two factors contribute to ultrafiltration. What filters out of the blood in the glomeruli are small site molecules, that is urea, salts, water, amino acids, glucose. The large-sized molecules in blood do not filter out, and these large-sized molecules are the blood vessel, the blood cells, and the plasma proteins. They do not filter out because they are large-sized. Whatever that now we have at the Bowman's capsule, whatever that filters into the Bowman's capsule forms the glomerular filtrate. The glomerular filtrate. So henceforth, we shall be talking of the glomerular filtrate. What happens then to the glomerular filtrate? The glomerular filtrate, from the Bowman's capsule, it is drained into the coiled tubules, the proximal convoluted tubule. The role of the proximal convoluted tubule is to provide surface site for the reabsorption of all glucose. Remember, we are talking of reabsorption now because absorption of nutrients takes place in our ileum, in the ileum. Now, there has been filtration from the blood. Some substances are useful, they have to be reabsorbed back. All glucose has to be reabsorbed back because glucose is the main respiratory substrate. In any case, even if we have excess glucose in our cells, it is converted to glycogen for storage, so there is no harm. So all glucose is reabsorbed. Where is it reabsorbed? At the proximal convoluted tubule, to ensure maximum reabsorption of glucose. At the proximal convoluted tubule, the tubule is adapted to ensure maximum reabsorption of glucose by the following ways. One, it is long and coiled. That provides a large surface area for reabsorption. Maximum reabsorption energy will be used, and therefore the cells, the proximal convoluted tubule has to be lined with the cells with numerous mitochondria to provide energy for active reabsorption of glucose. We can also borrow some adaptation from the ileum because the ileum also is a service for absorption, exchange of substances. The microvilli. So the tubule, the linings, the inner linings of the PCT will also have numerous villi to increase the surface area for reabsorption. From there, the glomerular filtrate now will flow to the descending loop of Henle. In this descending loop of Henle, that is where we will have reabsorption of water. Reabsorption of water. Obviously, then, osmosis. The cells there are highly permeable to water. At the ascending loop of Henle, we will have the reabsorption of salt or ions. The cells in the ascending loop of Henle are highly permeable for the reabsorption of salts or ions. And the main ion in our body is sodium chloride, of course. So reabsorption of sodium chloride ions takes place mainly in the ascending loop of Henle. At the DCT, the distal convoluted tubule, that is where we will have controlled reabsorption of water and some minerals, but mainly controlled reabsorption of water. Why controlled? It will depend on the body requirements. If the body requires to conserve a lot of water, then at the DCT, you will have increased reabsorption of water. But if for instance, you take a lot of fluids, a lot of water, and therefore your blood has a lot of water that needs to be allowed to get out through urine, then at the DCT, there should be minimal reabsorption of water. Then we ask ourselves, does it mean the DCT has some adaptations that enable controlled reabsorption of water? Yes. One, just like the PCT, the DCT is highly coiled and long and highly coiled. Long and highly coiled increases the surface area for the reabsorption of water, which means any mammal, like now the freshwater mammals, for the freshwater mammals, their bodies are susceptible to be having a lot of water because they are in an aquatic habitat, and therefore they require to lose excess water. To lose excess water, to allow the excess water get out in urine, then their distal convoluted tubule has to be short and less coiled. Short and less coiled, or not coiled, in order to reduce that surface area. In that means there will be a decreased reabsorption of water. Most of the water will be left to get out as urine. Those freshwater mammals will be producing high amounts of dilute urine. But if we take a desert mammal like the desert rats, in a desert, there is water shortage. And therefore, these mammals in a desert require to have maximum reabsorption of water at the distal convoluted tubule. And therefore, their distal convoluted tubules, as an structural adaptation, will be very highly coiled and long, highly coiled, to increase the surface area or to maximize the reabsorption of water. That is why those mammals in the desert will produce low amounts of highly concentrated urine. Now, by the time we get to the distal convoluted tubule, water has been reabsorbed, all glucose has been reabsorbed, ions and salts have been reabsorbed, and if there were any other useful substances like the minerals, like the vitamins, they could have been absorbed, especially at the PCT, which means the filtrate that is draining now to the collecting tubule, that filtrate that will be draining now to the collecting tubule will be having largely urea. No glucose, no vitamins, because they have been reabsorbed. And now the filtrate becomes urine, which is then drained, as we saw, by the ureter to the urinary bladder for temporary storage before excretion. Let us mention something about physiological adaptation of the kidney to the excretion of urine depending on the amount of fluids we take. And that brings in the role of the hormone ADH. antidiuretic hormone. ADH hormone secreted from your pituitary gland and has an effect on the distal convoluted tubule. The role of ADH is to increase permeability of the distal convoluted tubule to water. So let's say it is in the morning, you've taken a lot of fluids, like during break time, you've taken a lot of fluids, a lot of tea, and after a short while, you are run to the washroom to excrete. In another instance, you've taken meals, especially salt meal, and therefore your blood is likely to become somehow hypertonic because of increased concentration of solutes, the salts. You are likely to excrete less urine, which is highly concentrated, and that is the time normally the urine will have some kind of smell and some tinge of color. Why? When you take a lot of salt food and less fluids, your blood becomes a bit hypertonic. Once the blood becomes hypertonic, the role of our nephron now in osmoregulation and ionic balance comes in, and in this case, osmoregulation. Since the blood is hypertonic as a result of high intake of salts, the pituitary gland becomes stimulated. It releases antidiuretic hormone. Once the antidiuretic hormone gets to the distal convoluted tubule, the distal convoluted tubules becomes more permeable to water. As a result of being more permeable to water, there will be increased maximum reabsorption of water in order to dilute the blood to lower the osmotic potential of the blood to normal. That leads to production of low amounts of highly concentrated urine. Unlike when you take a lot of fluids, if you take a lot of fluids, then you need to allow a lot of the water to get out as urine, and that means at the distal convoluted tubule, there should be minimized reabsorption of water. And to guarantee minimized reabsorption of water, then the tubules should be less permeable to the water, and therefore the pituitary glands are less stimulated. No secretion of ADH or less ADH secreted. That forms the basis of the functioning of our kidney nephron, and it gives us a picture of when someone goes for dialysis. When someone goes for dialysis, which casually we talk of cleaning of the blood, it means it is the kidney nephron system, the process of ultrafiltration and reabsorption, that is not efficient in their body. I hope that is an exposition that demystifies to us the functioning and structure of our kidney nephron. For more details, I invite you to get in touch with our top-notch press. Get these very educative charts, like the chart on our kidney nephron, which captures the structure, adaptations, and the functioning of the kidney nephron. It has been nice exposition. Until next time, from Top Notch TV series, your favorite living organism, Mr. Bernard Osoro. Remember to subscribe to our online YouTube channel, Top Notch TV. Bye.